My name is Phil Gold, and I'm delighted that you're here, and thank you for coming. So, ladies and gentlemen, I have the pleasure actually tonight of introducing someone that I've known for a very long time, as a colleague and as a friend, and from whom I take great pride, not enough, from Marcel Behr. Marcel started his life in university at the University of Toronto. He survived that, got his bachelor's degree in biochemistry, and then went on to medicine at Queen's University. Am I better now? Okay, I'll start to echo in a moment. He obtained his MD from Queen's and then happily decided to come to McGill to do his residency in medicine and subsequently a fellowship in microbiology and infectious disease. At the same time, he completed a degree, a master's degree in epidemiology, which, as most of you know, is the study of the distribution, the incidence of diseases and how they spread in the population, the factors that allow these things to occur and how to stop them, and hopefully how to cure them with time. After completing his residency in microbiology and infectious diseases at the Montreal General McGill University program, he went on to Stanford University, where he did a degree, or his work, his postdoctoral fellowships in epidemiology, again, molecular epidemiology and bacterial physiology, becoming particularly interested in certain forms of bacteria called mycobacteria, which is the family to which the tuberculous tuberculosis bacterium belongs to. And you'll hear more about that family from Marcel this evening. He returned to Montreal McGill University, where he is now professor of medicine and the director of the McGill TB Center, as well as the microbiologist in chief at the McGill University Hospital Center. His life is threefold. He does teaching, he sees patients, but primarily he's involved in research looking at, in fact, the mycobacterial family, how they affect individuals in a variety of ways, and the one we're going to hear about tonight, of course, is the one dealing with Crohn's disease. Marcel is funded by the Canadian Institutes for Health Research for the research that he does, has been recognized by the Quebec government as a chercheur national, by the CIHR of Quebec, devoted à la recherche en santé in Quebec, and internationally. He has been elected to a very prestigious organization called the American Society for Clinical Investigation. But besides all of that, Marcel is a superb jazz pianist, which I haven't heard him do for a while. Ladies and gentlemen, Marcel Behr.
All right, thank you for the introduction, and I just want to make sure we're at the right microphone setting. Are you getting feedback there? Okay, because I understand we're being taped. So before I give the lecture, I want to foremost thank Doug Ream, who's here, for organizing the event and inviting all of you to come here. Doug and I discussed this over wine during a hockey game about 3 weeks ago, and I never would have thought that the Habs would still be in the playoffs now and that this many people would come out tonight. I'd also, of course, like to thank Dr. Gold for coming tonight and being here, and he'll be moderating any questions later on as the discussion evolves. But most importantly, I'd like to thank the Canadians and the Rangers for not playing tonight and leaving one evening that otherwise you guys would all be watching the game tonight. So, in this lecture, I'd like to just sort of outline my goals for the lecture. People come to lectures for different reasons. Everybody has their own background and they have their own interest. And I want to sort of clarify to you that I am both, as Dr. Gold said, I'm both a doctor and a scientist. So when I'm a doctor, I'm an infectious disease doctor. I prescribe antibiotics. I ascertain that people have an infectious disease, and then I provide directed medical advice. But when I'm a scientist, I'm exploring ideas that I hope will contribute to the changes in care in the next years, hopefully not many years to go. So clearly, as you understand, these missions are on a continuum. Medicine is, after all, an applied science, and our research of today aims to inform tomorrow's treatments and to revolutionize, if we can, tomorrow's treatments. Unfortunately, I am in a situation where I know some of you may have Crohn's disease or may have family members with Crohn's disease or children with Crohn's disease, and I would love to be able to tell you how to treat your children. And I would love to be able to tell you that there's a new treatment around the corner, but I am not in a position tonight to do so, in part because the data that we need to make firm clinical judgments and recommendations are not there yet. And these are the kind of data that I wish to secure by doing the kind of research I'm describing, so that we will be in a position to inform people in the future that there are actually data-driven recommendations and not just good ideas that haven't been substantiated through formal clinical trials. So I understand that you might want to ask me about medical advice, but it's just a time that we need more information. Everybody comes to science and comes to a room like this with their own biases. I'd just like to let you know my biases is that my research is funded by government agencies, by both Canada and Quebec, and in the past from the Crohn's and Colitis Foundation of Canada. And my funding has generally been to do research on how bacteria cause disease. I do not have pharmaceutical funding to my lab, lest you think that there's some big giant corporation that's funding the message you're getting tonight, and that's because most of my research is on tuberculosis and leprosy, and these are diseases of poverty. So I haven't had the pleasure of having pharmaceutical companies trying to seduce me. They don't bother, and I don't have to even come up with a judgment of whether to take the seduction. So the overview of my talk is I'd like to very briefly go over one of the prevailing ideas that I've heard over the past few years about Crohn's and autoimmunity, and I'm going to then tell you how we can now flip this in a different direction and ask if Crohn's, in fact, is not due to aggressive immunity, but insufficient immunity. I'll tell you some of the work we've done to contribute that, and then I'm going to tell you how that could perhaps point to mycobacteria having potential role in Crohn's disease before concluding. So Crohn's as an autoimmune disease. I wonder if anybody in the room has been told that Crohn's is an autoimmune disorder. Please raise your hand. Okay, so I have been told that as well. So you're not imagining it. I have written consent forms to try to do research on Crohn's disease, and the consent form came back with a first sentence: you have an autoimmune disease called Crohn's disease. And a few years ago, I stepped back and asked myself, how do we know this? In fact, I'm a skeptic, right? So I often ask, how do we know anything that we really think we know? Around that time, I even went to Dr. Gold's office and I asked him, what are the autoimmune diseases? How do we know any autoimmune disease is an autoimmune disease? And we looked around for the evidence. And as some of you may know, in much of medicine, we have these standards to say something causes something. And in infectious disease, they are called Koch's postulates. You say the bacteria was there, we got it from the animal, we put it into another animal, the disease happened, and that's how we all agree that a bug causes disease. In the case of autoimmune diseases, a scientist named Witebsky postulated in 1957 that there were 3 preconditions for deciding that some disorder is an autoimmune disease. And the causal model that he proposed was that you can direct antibodies in the blood of the patients with that disease, and these antibodies are directed at self. And once you know that there are antibodies against self, you can even find the molecule in the body that these antibodies are directed against, and these are called the antigen. And that if you put that antibody and that antigen together into an experimental model, together, they will recapitulate the disease. So for instance, some aspects of some autoimmune diseases, these have been fulfilled. If you take the antibodies from Wegener's disease and the antigen that they recognize and you put them together, there are animal models where you get the kidney disease of Wegener's disease. So we asked ourselves, had Crohn's disease fulfilled 1, 2, or 3 of these postulates that would make it an autoimmune disease? And the answer we found by looking in the literature and looking hard and looking over and over again was, were there antibodies in the blood directed at self? Well, no, not yet. A minority of patients have some autoantibodies in Crohn's disease, but not really much more than a control group would have. Well, then the second part here, is there a self molecule that's recognized by those antibodies? Well, if the antibodies don't exist, it's pretty hard to find the self antigen that the non-existent antibodies are allegedly recognizing. So yes, there are some antibodies that are found in up to 25 or 30% of Crohn's patients, for which we know the self molecule is in the pancreas. But that's in a minority of people. It's not a diagnostic test, as many of you know. There is no blood test for Crohn's disease, and if you are suspected to have Crohn's disease, nobody orders an autoimmune antibody panel that secures the diagnosis. So therefore, if part 1 had not been met and part 2 depended on part 1, then you can imagine that if you put the 3 together and say this antibody and antigen put together result in the disease, this condition had not been met either. Now, we weren't really sure as infectious disease doctors, so I equipped myself with an immunologist and a keen graduate student, and the 3 of us scoured the literature because we were afraid that we were going to say something really wrong. And when we submitted it to The Lancet, which is a relatively recognized medical journal, and said there's really no evidence this is an autoimmune disease, we were quite surprised that 3 reviewers who we didn't know, we didn't even propose reviewers, they all agreed with us, but they just said, could you change the references because they'd like to see their name in print rather than somebody else's name in print. And since that article, actually, I haven't had anybody tell me what was wrong with our argument in the next 4 years. So this is not a new idea that I'm proposing today. It's at least 4 years old. So it is my view that the notion of Crohn's as an autoimmune disease has not been directly proven in any manner. So why do we think it's an autoimmune disease? Because we use anti-inflammatory treatments, and these anti-inflammatory treatments that are used to treat Crohn's disease have some anti-immune basis, but they're really anti-inflammatory, such as steroids and anti-TNF. So is that sufficient to say a disease is autoimmune? As an infectious diseases doctor, I would say it probably is not sufficient. I can tell you that we use the same sorts of drugs when we treat certain infectious diseases. If you take bacterial meningitis or if you take tuberculosis meningitis, we give antibiotics, but as adjuncts, we give steroids to reduce the inflammation. It's not the bacteria in your head that are going to kill you from bacterial meningitis. It's the inflammatory immune response to the bacteria, and that's why steroids are life-saving in these conditions. In the case of leprosy, there are certain reactions to this bacteria that benefit not only from steroids, but there are actually published papers where people are using the exact same anti-TNF agents that are used in Crohn's disease to treat their reactions to leprosy. So it is my view that there's no direct evidence of autoimmunity. There's only indirect evidence in that we use these sorts of agents to treat the disease. So what about the alternative? Rather than Crohn's being due to an overtly aggressive and unnecessary immune response, how is it possible that I'm going to suggest that maybe because there's a deficient immune response? Has anybody in the room been told Crohn's is an immune deficiency? And thus, fewer hands go up. So what is immune deficiency, and what would be the evidence that's consistent with immune deficiency in Crohn's disease? So classically, when I was training at the Montreal General Hospital and the head of medicine was this immunologist named Dr. Gold, we always thought that immune deficiencies were children who had many, many infections due to many, many bacteria over and over again. So if you're immune deficient, you'd get strep throats and ear infections and pneumonias. Or at that time, we didn't have antiretroviral therapy. So the HIV patients, they had full-blown AIDS, they had no CD4 cells, and all kinds of different infections were being seen in them, from viruses like CMV in their eyes, to parasites like Cryptosporidium in their stools, to bacteria. And thus, we sort of thought of immune deficiency as these flagrant states that were best depicted in Hollywood by John Travolta in The Boy in the Bubble. So anybody here who's old enough to know that John Travolta acted in movies before Pulp Fiction, one of his early movies was this idea of an immune deficiency state where you're just completely intolerant of any bacteria in the environment. There is a more subtle and nuanced definition of immune deficiency that is now emerging, which is considered very selective, that you could have defective immunity to only one pathogen. And in the absence of that pathogen, you wouldn't know you're immune deficient. And how would that happen, that I could be immune deficient to something? Well, it's 2014, and let us postulate that I am not able to handle the plague. And if I was in 1350 in Europe and the plague hit me, I would be one of the 3 people in Europe who died of the plague. But in 2014 in Montreal, I'm not going to meet that bacteria. So I will just go along not knowing that I have a problem handling the plague. Same with cholera, the same with typhoid. So the idea that's increasingly emerging is that many of us have difficulty handling 1 or 2 or 3 bacteria, of which most of us are simply not encountering them. And that could even explain not only that immune deficiency can happen, cannot be diagnosed in some, but there's even situations where there are twins, where one person has Crohn's disease and the other person doesn't have Crohn's disease. How is that possible? Well, perhaps they both share an immune defect, but one has been exposed in a way that another has not been exposed. So this is the newer interpretation of immune deficiency. And this was published in a journal that has the word immunology in it. And again, it was peer-reviewed and vetted in the scientific community. So if we examine what are accepted to be immune deficiencies, then we can ask, does Crohn's meet the kind of definitions and the criteria that are seen in immune deficient states? And I'll go through 3 criteria. One is analogy. If it looks like a duck, it quacks like a duck, usually it's a duck. I'm going to tell you a little bit about clinical immunology that is not my work, but I will share research that I've read about that I find is quite provocative, and then I'll tell you about genetics. And the beautiful thing about genetics is that we know that DNA changes happen before the disease. So it's one of the few types of studies we can do where we can yeah, I'm going to get rid of this, where we can really ascertain a directionality. Your mutation happened before the disease. All the other studies, it's very hard to disentangle the two. So, analogy. There are 2 well-established immune deficiency states that are seen in immunology clinics. The first one, chronic granulomatous disease, is seen primarily in pediatric clinics. And in this study from Steve Holland's group at the NIH in Bethesda, Maryland, when they looked at 140 people who had chronic granulomatous disease, which is a card-carrying established immune deficiency, and they did biopsies on their gut, 46 of the 140 had biopsies that were indistinguishable from Crohn's disease. So did these children have Crohn's disease, or did they have chronic granulomatous disease? Many of you know that Crohn's disease is defined as an idiopathic disease. You define that you have Crohn's disease because you've ruled out other things. So once you've shown they have this, it is not Crohn's by definition. In fact, some of the children had been diagnosed with Crohn's disease, and then they had special neutrophil studies done. And when the special neutrophil studies were done and were showing impaired neutrophils, they were effectively rebranded from CD to CGD. This has also been described for something that you probably see in your own clinic, common variable immune deficiency. And in this study, also about 40% of people with an established, conventionally understood immune deficiency have pathology that looks like Crohn's disease if you look in their gut. So that's analogy. It doesn't prove anything, but it provokes you to think. It generates hypotheses. These are the studies by Tony Siegel's group in London, England. That first disease on the previous slide called chronic granulomatous disease, he discovered the defect in chronic granulomatous disease back in the 1970s, and he went on to say he sees the same type of defect in Crohn's disease. Nobody listened to him, so 30 years later he went back and he started to design very novel assays to look at Crohn's disease patients and how their immune system responds to trauma and bacterial challenges. In the first one, he said Crohn's patients get biopsies. Some of you have had endoscopy, or somebody in your family has had endoscopy. And what he did is he said, given that people are getting biopsies, why don't we biopsy the biopsy site half an hour later, and see how well do people repair the biopsy site. And he looked at Crohn's patients, and he looked at colitis patients and people who were healthy controls or who had other reasons. And he found that in the Crohn's disease patients, they had defective recruitment of cells, defective recruitment of these things called cytokines that indicate that you are responding to that trauma. He followed that up with quite an ingenious study where he took bacteria and he radio-labeled them, and he put them killed but radioactive into the skin of an arm. And then he did something called a gallium scan, where you take the white blood cells of the body, you label them with gallium, and you put them into the vein in the other arm. So he had radiolabeled bacteria in one arm and radiolabeled white cells in the other arm. And he said, how long does it take for these white cells to go find the bacteria and clear the infection? And he found that the Crohn's patients, compared to the control groups, had less recruitment of white cells to the infection and delayed clearance of that bacterial stimulus in the other arm. So that's clinical immunology. Again, you're always going to say, well, is it because they have Crohn's disease that's deficient or because they have immune deficiency they have Crohn's? It's very hard to disentangle the two. So then there's genetics. If you look at the genes that are being uncovered that have been linked to Crohn's disease, many of them, when you look on them at the first pass, they're involved in some sort of immune response to bacteria. So that tells you there's immunity, but it doesn't tell you if it's more or less. However, if you look at the mutations that predispose to Crohn's disease, so you have the wild type, if you like, the reference allele, and then you have the variant that leads to a higher risk of Crohn's disease. The variant that's Crohn's associated is often a loss of expression or a loss of activity. In the case of the NOD2 protein, the protein is broken and it doesn't do its job. It does not respond to bacterial stimulus. These are not my quotes. These are articles from the first three and most predominant or preeminent Crohn's disease genes as they were published in the journals Nature, Nature Genetics, and Nature Genetics. So Ogura wrote, when they discovered a link between NOD2 and Crohn's disease, these data suggest a link between immune response to bacterial components and development of disease. Hampa wrote, when they discovered this gene encodes a protein in the pathway that processes intracellular bacteria. And Parks wrote, taken together, the evidence regarding this gene plus those genes up there strongly implicate defects in innate immune pathways and handling of intracellular bacteria. The geneticists wrote this, okay? So what do you do with this information? You start to create patterns, you start to look for associations, and you try to figure out how you can then mechanistically dissect these. But we know that these things predispose to Crohn's disease. Unlike the other one, you don't know if it's because they have Crohn's, their immune response is down. These ones, you know, you have these mutations at birth. A couple of years ago, with a graduate student, Jean-Daniel Lalande, we were asked to write a review of some of these ideas. And we did this very naive, what's called the Venn diagram. We just chose a few of the genes that were out there in the literature. And we said, there are human genetic studies that describe genetic variants that predispose to Crohn's disease. And some of them are here, ATG16L1, NOD2, IRGM. These are ones that were on the previous slide. And there are also the same types of genetic studies that you take people with tuberculosis and you take their family member who didn't get tuberculosis and you say, why is it that everybody in the family was living together and some people got disease and some people didn't get disease? And you say, well, there's vitamin D receptor and there's this thing called NRAMP that was discovered in Montreal. And there's actually TB genetic studies that say NOD2 variants predisposed to TB and IRGM variants predisposed to TB. And then there's this other mycobacterial disease here called leprosy, which we don't see very often. And you look at what are the kind of genes that have been associated with leprosy. And you see that this NOD2 gene has also been associated with leprosy in multiple studies. And this one here has been seen in Crohn's and leprosy, and this one here has been seen in TB and leprosy. Now this was really just taking some literature out there and sort of saying, we think we see an association. It was done much more definitively by Judy Cho's group at Yale a few years ago, a few years later, where they did what's called a genome-wide association study. They did many of them, and then they put them all together. And this is an analysis of 75,000. I'm going to come back to NOD2 later. This is an analysis of 75,000 Crohn's patients, IBD patients, and 75,000 controls, published in the journal Nature, genome-wide association study. This is very complicated. The list of authors would go through the floor here. Many, many groups contributed to it. And they said, what are the patterns we see in the IBD genetics? And this is their writing, this is not mine. We observe a striking overlap between susceptibility loci for IBD and for mycobacterial infection. They went on to say the genes shared between host responses to mycobacteria and those predisposing to IBD. So the geneticists are saying Crohn's genes are involved in bacterial handling, and it looks like they're involved in mycobacterial infections. So, analogy, check. Immunology, check. And the genetics says there is defective handling of bacteria. That would be an immune defect. Which bacteria out there among all the bacteria on the planet could exploit these sorts of immune defects to cause a chronic inflammatory disease? Well, simplified speaking, when we look at all the bacteria, they are named from Christian gram to be gram-positive, and they stain dark blue, or gram-negative, and they stain pink. So Staphylococcus, which is in your nose, is a classic gram-positive bacteria, and E. coli, which is in your digestive system, is a classic gram-negative pathogen. In the gram-positive group, there's a very unusual club of bacteria that are called mycobacteria. They are known because they have this very fatty cell wall that's made of things called mycolic acid. You can think of this as a bacteria that's sitting in a tub of margarine or something. It's very waxy, and it's a very unusual bacteria. It's considered gram-positive, and it is known for the agents of tuberculosis and leprosy. And that itself reminds me of analogy, because tuberculosis is a chronic inflammatory granulomatous disease, and leprosy is a chronic inflammatory granulomatous disease. So they're already, if you were a profiler, the kind of bacteria that would cause chronic inflammatory granulomatous diseases. Also, these are intracellular bacteria, which mean they live inside of our cells, inside cells, white cells called macrophages. And that's important because the NOD2 gene is expressed as a protein inside of the cell. NOD2 is an intracellular receptor that recognizes bacteria that are in our cells. It doesn't recognize bacteria that are free-floating. So we asked two questions, very simple questions. Is NOD2 important for mycobacterial infection? So the answer is yes. And how did we do it? We obtained mice that were disrupted for NOD2, so they do not have NOD2, and we challenged them with the bacteria that causes tuberculosis. And we asked, how do these mice do on day 1, on day 7, on day 21, and up to 6 months? And when you take their cells out of them and you throw tuberculosis on, their cells are less responsive. And when you look at 14 days, and you put bacteria into their lungs, and then you take the fluid out of their lungs, their lung fluid is less responsive. And if you look at 28 days and you say, how well do they make antibodies or cell-mediated immune responses to tuberculosis, they're defective. And if you go out to 6 months, the NOD2 knockout mice had more bacteria, and they had more aggressive disease. So what you have is early, you have impaired responses, and late on, you end up with inflammation because you have an uncontrolled infection. So that's nice, and people in the room can say, I bet you could have done that with any bacteria. There's nothing in that paper that tells you it's about mycobacteria. It just happened to be the bacteria you used. So we followed up with a different question, which is NOD2 specific for mycobacteria? And here the answer is yes. We use something called genetics squared. I'm not going to give much detail. It's all published now. In this paper here, the mice either have NOD2 or don't have NOD2. In this paper, we also made a mutation in the bacteria. So the bacteria either had the mycobacteria cell wall or they had the more standard staphylococcal cell wall. So we had bacteria that had this mycobacteria special molecule, or mycobacteria that had a normal cell wall. And we found that only when the mycobacteria made that special molecule was NOD2 important. When we disrupted that molecule, NOD2 was unimportant. When we put the molecule back, NOD2 was important. What do I mean by this molecule being special? The cell wall of a bacteria is recognized by the immune response, but some molecules are more potent than other molecules. What is potency? I can give you an ounce of alcohol and an ounce of a drink, and you'd say, what's an ounce of a drink? If I give you an ounce of light beer, you're not probably going to get drunk. If I give you an ounce of vodka, it's still an ounce, but there's a lot more alcohol in there. So if I take a microgram of the cell wall of bacteria, I give you the mycobacteria version, that's the vodka. The other bacteria version, that's the Coors Light, okay? So that it's a more potent activator of NOD2 that is made by mycobacteria. And we followed that up in 2 papers, one that was published earlier this year, with 2 different mycobacteria. We've done it with 2 different grad students, and we've got the exact same results with some variants. So I think it's quite robust, and 2 other groups have also shown that this mycobacterial version is much more potent at activating the NOD2 response. So this is the way I conceptualize NOD2. If any of you are lawyers or judges, you can think of this as sort of the scales of justice. So this is a balancing platform here. And if you have features over here, it's going to tip this way. And if the features are here, it's going to say that NOD2 is probably important in handling and killing this bacteria. And if you're over here, it means that those features say you can probably deal with that bacteria without NOD2. It won't matter if you do or don't have NOD2 if you're over in this column. Okay, do you understand the balance here? So what are the things that we now know from the literature after about 10 years of studies of NOD2? NOD2 is much more important for gram-positive bacteria than it is for gram-negative bacteria. There are many papers by many independent groups looking at different bacteria. There's very little associating the need of NOD2 to handle a gram-negative bacteria. NOD2 is inside of the host cell. NOD2 is more important for bacteria that are inside the cell than extracellular bacteria, which are on the outside. If the bacteria is on the outside and the protein's on the inside, they're not going to get to know each other too well. So the absence of NOD2 is not going to be as important for extracellular bacteria. And in our work, we showed that if you make the vodka version of the bacterial cell wall, it's much more important that you have NOD2 than if you make the light Behr version, which is much less potent. If you were to now say, what is the profile of this kind of a bacteria? It's a mycobacteria, and what is the profile of this kind of bacteria? It's E. coli. So the collective literature from a number of different papers suggests that NOD2 is important for such infections and less important for such infections. So, immune deficiency and mycobacteria. To this point, I think I have hopefully introduced you to the notion that the data point to a problem with the recognition and killing of mycobacteria. Not in everybody, but certainly in the people who have NOD2 mutations, which could be responsible from 20 to 30% of Crohn's patients, and perhaps other genes that are on the NOD2 pathway. I've told you that I did my work with tuberculosis, but of course you know that tuberculosis is a lung disease, so you're asking, well, why would this problem here lead to intestinal disease, and which mycobacterium would you be concerned about? This bacteria is not tuberculosis. It's called Mycobacterium avium paratuberculosis, and I will simplify it as MAP. So this bacteria MAP, once again, we can ask ourselves about analogy. What does this bacteria do where it dwells? It causes inflammatory bowel disease in cattle and in sheep and in goats. It causes a chronic granulomatous inflammatory bowel disease of these animals. Those of you who do not have a cow in your house or in your neighborhood here probably don't see a lot of them, but you're not supposed to see the ribs. You're not supposed to see those cavities. This is an emaciated cow, and it causes poor productivity, weight loss, and diarrhea. Diarrhea would be what we would recognize as sort of a medical symptom. Weight loss is something that you see by weighing the cow, but in fact, the first sign that the farmer will have that they've got MAP in their animal is that their cow is consuming more food than the cow is producing in milk. The cattle are not here as pets, they are economic, and the driver of all MAP research is the economics of whether this animal is producing more in milk than is consuming in feed. We know MAP causes disease. This is called Johne's disease in cattle by experimentally infecting animals. This is what I called Koch's postulates. You put in the bacteria and you wait. But we also know you have to wait for 3 to 5 years. And we know that in most cases, a PhD takes less than 5 years. So you can't do a lot of research on a 5-year model. You give your PhD student the bacteria and you say, infect a cow, and 5 years from now, write a dissertation, and you're not going to get a lot of experiments done. Because if the first experiment doesn't work, then they're there for 10 years, and there's no funding. So you can do descriptive studies when you've got a disease that takes 3 to 5 years. It's very hard to do what we call mechanistic studies. You can't change the course of disease, speed it up, slow it down, reverse things if the natural disease takes 5 years. If you don't want to look at this cow anymore or imagine what Johne's disease looks like on the inside of the cow, I recommend this fantastic resource that only I can offer you, which is this. The disease book called Paratuberculosis Organism Disease Control, edited in 2010. Now, why did Des Collins and I write this book? Largely, we wrote this book because there was a lot of information about MAP on the internet, and there was also some misinformation about MAP on the internet. There wasn't a textbook of paratuberculosis since 1913. So in the intervening 97 years, there were a lot of things that were being said about the disease, and I was having a hard time going back to the primary literature and finding out what exactly do we know and what do we think we know about this disease. You've all heard Donald Rumsfeld teach you about the difference between known knowns and known unknowns and unknown knowns, and I wasn't able to sort it out. So we expeditiously decided that if we asked 30 people who really knew their field of paratuberculosis to write a chapter, we would get them to do the literature review and tell us what they knew about it. So there are some statements in there that startled me when I was editing it. Anybody here who's read about MAP on the internet has been told or has read somewhere, I have read it many times, it's an emerging threat and the prevalence of MAP is going up, up, up wherever people look. And the person who wrote in that chapter on the prevalence of MAP said, the only way we can know if it's an emerging threat and the prevalence is going up is you have to go to the same place, use the same test, the same sampling strategy, and do the same study over several different years. And it's really boring in science to say, I did a study in 1985, I'm going to go back there in 1995 and do the exact same study. And then to get somebody to fund you to go back in 2005 to do the same study a third time is unlikely. And if you're still successful, there's no way they're going to let you go there in 2015. So he said, in theory, those would be the conditions that we could say MAP is growing. And he could not find a paper published by anybody had used the same sampling strategy, the same test, the same place over time. It might be going up, it might be going down, we don't know. So there's a lot of things that we think we know about MAP, there's a few things that are true about MAP, and the idea in this book was to try to assemble it, and if only to say what we don't know, at least we can list the things that might be experimentally testable. So is this a bestseller? No. There are not a lot of people who've bought this. In fact, not many people buy textbooks at all in 2010. You can get this on Google Books. So if you go and download whatever you want to read on Google Books, I won't get any royalties. It doesn't matter. At least the record is more or less established for what paratuberculosis does and what paratuberculosis doesn't do. What do we know about MAP in Crohn's disease? We know that the DNA of that bacteria has been detected in biopsies of Crohn's disease much more commonly than in biopsies, for instance, of ulcerative colitis. If MAP was merely seeping into a biopsy, you would think it would go into all kinds of forms of IBD, but we see it in Crohn's biopsies much more likely than ulcerative colitis. And when this was assembled over 28 studies, the odds of finding MAP DNA by this PCR test in Crohn's is about 7-fold enriched compared to other tissues. In our lab, we used this very old-fashioned thing called a microscope. First, we measured mycobacteria in mice, and we saw how long they were and how short they were and how fat they were and what shape they were. We tried different ways of dyeing them and staining them, and then we applied them in 2 samples from Montreal and from Hamilton with 2 readers, and we found that these mycobacteria were 8.5 times more likely to be seen in Crohn's tissue than in control tissue. And then Ingrid Olsen did this fantastic study in Oslo a few years ago. She came from a lab that had learned how to work out why we have a disease called celiac disease. Some of you may have heard of gluten enteropathy or celiac disease. And that lab had figured out how to biopsy the gut in celiac disease to grow the lymphocytes in the lab, the white cells, and to have individual clones of white cells growing in the lab so you could challenge those clones of white cells in the lab and find out what exactly are they recognizing. She applied that exact same technology to Crohn's disease and was able to establish that this clone of lymphocytes recognizes exactly that mycobacterial antigen, and this clone recognizes exactly that mycobacterial antigen in a way that differed from her control group, which was ulcerative colitis. She followed it up and said, in fact, the amount of lymphocytes that recognize mycobacteria in Crohn's is the same amount, if you like, as the amount of lymphocytes that they see in celiac disease that recognize the celiac antigens. So that's association again, right? Remember I told you genetics gave you direction. This is association, okay? And so the problem with this kind of literature is we see A and B. You cannot say A led to B any more than B led to A, or there could be some unknown feature C that predisposed to both A and B. So what can you do to try to see if really A did lead to B? Okay. Well, one way you can do infection to disease studies is the classical way. Some of you may or may not know that we've established that E. coli can give you traveler's diarrhea. And Helicobacter can give you peptic ulcer disease, and Campylobacter can give you gastroenteritis by putting these bacteria into human volunteers. Those were all done either in the early 20th century on volunteers that would not pass today's ethics committee, or even in the late 20th century in very controlled settings where you could take even E. coli and mutate one gene in it, and it didn't give people diarrhea, and you put that gene back and it did give people diarrhea. And that can be done in human volunteer studies because these are bacteria that give a short-term disease for which there's a cure. So even if all else goes wrong, you can clear the infection. This cannot be done, to my knowledge, with MAP. I do not think we can take vials of MAP cultures from my lab and give it to hundreds or thousands of volunteers and say, what is going to happen? We know in cattle we'd have to wait 5 years. In humans we might even have to wait longer than that. I do not think we can give a bacteria to people in the intent of causing chronic disease, especially in a setting where there is no currently approved cure for that infection. So we wouldn't be able to eliminate the infection, so if all things go wrong, we might get disease, but we'd have a lot of sick people who would be suing McGill University or the MUHC. So I do not think the human volunteer model can be used to establish causation. So another option that we have opted for more recently is to take mice that are disrupted for known Crohn's genes. Some of them are things like NOD2, but we have other mice that are disrupted for other genes that mean they have very impaired immune responses to mycobacteria. And what does that enable our laboratory to do? We can control the bacteria. We can put in MAP, or we can put in other bacteria. We can control the time. We can look in weeks, we can look in months. We can change the dose. We can do all kinds of things. We can mechanistically play it out. And if we can do things in weeks and months, then you can imagine that grad students and postdocs can actually see results and act upon it in a way that you cannot do if you infect a cow and you wait for 5 years. So then what can we do? If we do those infections, we can monitor outcomes. And in our lab, we have now validated 3 robust ways of studying the outcome of these MAP infections. We can count the bacteria. We have validated a way to count how many MAP bacteria are in a milligram of tissue, and we can see how many bacteria are in the intestine, in the liver, the spleen, the lymph nodes, in all different parts after an infection. We can count the cells that come and migrate into the tissue and ask, are there more or less cells that are inflammation causing? And we can fix that tissue and ask, do we see paratuberculosis pathology? Do we see the pathology that we would see in a cow? And is it similar or not to Crohn's pathology? It'll never be possible to say an animal has Crohn's disease because that would not be a mouse then. But you can say, is the same processes involved, right? We use mice not to model diseases. We use mice to monitor processes. And we can say, do we see the types of pathologic responses that we would see in the humans that we wish to study? And a mouse model of that nature, to me, has two conceptually different utilities. One is it allows us to ask scientific questions. We can ask about causation because we can put in the bacteria before the genetics. We can put in a second bacteria, or we can kill the bacteria, or we can challenge twice and see what happens. We can have a vaccine before the bacteria. There's all kinds of things we can do because we control the experiment, we assign the directionality, and we can perturb the system one way or the other. And that would be what you would consider more or less basic fundamental research. And people in this room are probably not that interested in that basic fundamental research because you may be sick, you may have a family member or a child who's sick, and you don't want to know about the mechanism of that sickness. You want to know, is there a translation? Is there a product that could come out of a model? And I believe that this model can offer translational advances. In such a model, you can ask about the potential benefit of different treatments. You can give steroids. You can give anti-TNF. You can give antibiotics. If you don't like these antibiotics, you can give those antibiotics. If you like the anti-TNF, but you'd like to try antibiotics, you can give both, right? There's no reason you can't give antibiotics plus anti-TNF. We needn't say that antibiotics are the opposite of anti-TNF. In fact, we know from diseases like leprosy, like tuberculosis, and like even bacterial meningitis, that we often give antibiotics to kill the infection, and we give anti-inflammatories to temper the immune response. It's important, though, when you do these sorts of things and you look around the internet and say, well, which treatments do we want to do? I think it's not just semantic to define quite deliberately what we want to do in such translational studies. We do not set out to prove that something works. That is the worst thing you can do in science. You say, I know this is going to work, I'm going to try it, and if it doesn't work, I'm going to try it harder. And eventually we know that that's the definition of insanity, which is to keep trying something and hope that the outcome changes. We do not prove that antibiotics help. We investigate whether antibiotics work. And if the antibiotics we try don't work, we can cross it off the list and we can move on to something else. It may be that this is an infectious disease that benefits from some anti-TNF, and that some of the antibiotics help, but others don't. And we cannot go in there thinking that we know ahead of time what will and what will not work. But the beauty is that our model is enabling, because we don't have to wait 5 years for these results. We can now do experiments that happen over the course of several months. And after 3 months, you can reassess, you can reboot and say, oh, let's try it in a different way. And at least you have an iterative process where you can try to improve your method with each passing way. So in conclusion, I do not consider that there is compelling evidence that Crohn's disease is an autoimmune disease, and that is something that we've written up and published, and I'm sure it will be continued to be said to be an autoimmune disease. I'm not convinced that that is the primary driver of the disease, and rather I think that primarily there may be an immune deficiency, and that immune deficiency is enabling of something that is effectively an infectious disease that drives the pathology. And the immune deficiency meets analogy, immunology, and genetics, and I think is consistent with the microbiology. Which bacteria could exploit this immune deficiency is an area that is an active field of investigation. I think the data are consistent with mycobacterial infection. The science is incomplete, and we, I think, are in a really good position to advance the science and maybe provide some answers relatively soon in this direction. Finally, I would like to recognize a quote that I read a few years ago from Stephen Jay Gould, who was describing a revolution in thinking in his time as a paleontologist. And he was describing that when he went to graduate school, nobody believed in continental drift. He said there was no way in the world that Africa and South America could meet or unmeet, and without a mechanism, people kept on finding fancier and fancier explanations that the fossils on both sides of the ocean were the same, and they said maybe there were glacial bridges or skyhooks or birds were dropping the fossils, and it became harder and harder to explain that Ireland and Newfoundland looked the same. And then somebody came up with a mechanism and said, this is how continents move. It's plate tectonics, and it's not the continents moving, it's the shelf that's moving. And in that case, he said, in fact, all the people collecting all the data were trying to fit the data into the old model. And he said, new facts collected in old ways under the guidance of old theories rarely lead to a revision of thought. Facts do not speak for themselves, they are read in the light of theory. And that's why I believe that looking only for translational advances without advancing a mechanism may be short-sighted. It could be that we really have to understand the cause of disease to drive the research agenda in a new direction. And with that, I would like to thank the people in the lab who've done the work, along with the collaborators who work with us, the funding agencies who've supported my work over the past few years. And most importantly, I would like to thank Doug and Kim and acknowledge that they have gone out of their way to create a fund that is helping support the research that I'm describing, and it's much appreciated. So thank you, everybody, for your attention. I don't know if we're going to try this or... Okay.
I think at the end of the day, you've had a full course in infectious disease immunology and genetics. But if you're not in the field of microbiology, of molecular biology, biology in general, you want me closer here? Yeah. Okay. And at the end of the day, you think that this is actually something that you want me to crawl in? Thank you. That effectively you believe that this is something that's sort of straightforward. You have no idea what's happened in my lifetime in medical biology. What you just heard is about funny-looking molecules. When I began to look at immunology and in genetics, I was looking at funny-looking kids. So we've gone from whole people to molecules inside cells. The gestalt is totally different, but the way that one does research remains unchanged. So, with that said, let me ask for questions to Marcel from the floor on this enormous course you've had this evening. I think it's something you can take home with you and put in the bank. Yes?
Just to be clear, Marcel, in the model, when you're infecting the mice with paratuberculosis, they're coming down with Crohn's 100% of the time?
In the model, when we infect mice with MAP, we have infection. We find bacteria all the time. We have pathology. We see pathology that is consistent with paratuberculosis. So is that Crohn's disease or is that paratuberculosis? That becomes semantic. The animals have pathology in their small bowel, and we see it in 1 month, which I think is a lot better than waiting for 5 years. Can you take that slide and put it in front of a pathologist and say, is this Crohn's disease? The first thing a pathologist will say is, this isn't even a human. This is small tissue. So at what point do you say that the pathology is or isn't the same? That's a very difficult issue to study. I can also say that if you give a mouse tuberculosis, it's not the same pathology as a human with tuberculosis. So mouse models and these kind of infection models we use, I think I'm very careful to say they allow us to study processes. There are a lot of mouse models, and Dr. Gold can give you more information, where somebody has cured cancer. There are mouse models where somebody has cured Alzheimer's, Parkinson's, Huntington's, you name it. There's all kinds of mouse models of a disease where they cured the disease, then a drug came out, and that drug went to phase 1, phase 2, phase 3, and then it disappeared. So we always have to remember that mice allow us to model processes, and because we can genetically manipulate them, and we have immune markers, and we can study immune things, and we can and perturb the system, we can dynamically change the process. But at the end of the day, we have to be quite humble before all of the times in all of the diseases that a mouse model of something was cured, and as you know later, the disease was not cured. And you probably know many examples of that, for instance, in the cancer literature.
Unfortunately, yes. As soon as we have a cure for cancer in man, we're going to apply it to mice. Yes?
Have doctors ever tried to cure Crohn's in either children or adults with any type?
Sure. So there's two ways to give antibiotics to humans with Crohn's disease. One is to give broad-spectrum antibiotics, which is often a combination of something like ciprofloxacin and metronidazole, and there are clinical trials where that's shown benefit. There are also clinical trials where the ciprofloxacin metronidazole group was indistinguishable from the placebo. And I think the meta-analysis of that combination has shown that it may reduce the recurrence of disease after surgery, but it is not considered to be a primary treatment to put somebody who is sick into remission. Then there are also studies where people have tried these antimycobacterials. The problem with that disease I described to you, paratuberculosis, as I've learned by editing the book, is it is an economic disease. And therefore, there is no economic incentive for any farmer to keep a cow for 6 months and give that cow large doses of antibiotics. The dose of rifampin that we use when we treat somebody with tuberculosis is 10 milligrams per kilogram. Now, you ask yourself how many milligrams of rifampin you have to give to a cow, and you realize that the farmer is going to send the cow to the slaughterhouse. Okay? So there are almost no data on how to treat MAP infection in a cow. There are a lot of data on how to kill MAP on a petri dish, but we don't know if the cure for MAP in a petri dish is going to be a cure for MAP in an infected animal. So when people have done randomized trials, they said, well, why don't we treat with the antibiotics that seem to work in other mycobacteria, and we'll try this combination of this, this, and this, and we will hope it works. And that's something that could work. In a scientific sense, that's doing a phase 3 clinical trial without preclinical data and hoping that you got the right combination of the right drugs with the right dose and the right duration. In a study published by Selby in Gastroenterology in 2007, they did something like that, and they recruited about 200 Crohn's patients. Half got antibiotics, half didn't. And they found that after, 16 weeks, there was about a 16% benefit to the Crohn's group. And then over time, depending on how you analyzed it, either the benefit waned or the benefit remained, but it never got better than 16%. So is 16% 1/6 full, or is it 5/6 empty? The authors and the editorial concluded that there was a failure. They said that if it's an infectious disease, we should have 80, 90, 95% cure. 16% is a disaster. If you're looking at some of the immunologic agents, they're getting FDA approved with 16 to 20% benefit. So how you judge benefit depends on the window, the prism that you've predefined as benefit. Do we know if that was the right combination, the right duration, the right dose? Maybe they were onto a combination that could have given 40% benefit, but in the absence of that kind of preclinical data, it's really hard to give antibiotics on sort of, just lick your finger and hold it to the wind. And I think there could be a potential to actually start to really rigorously try different combinations and try to find the best combinations in a preclinical model before doing any such clinical trial in the future. That's my view of that.
Charles?
Is there any genetic link between the incidence of disease and, say, where a person lives, or perhaps the Jewish, often you hear that Crohn's is a Jewish disease? How many research into that?
Repeat the question.
Yeah, the question was, if I understand, is Crohn's a Jewish disease? Is Crohn's linked to where you live? So the answer is yes, the incidence is elevated in Ashkenazi, and I think there's some data in Sephardic Jews. I don't think it's because people live in Hampstead or Côte Saint-Luc that it's elevated, but rather that there's a genetic predisposition that goes with their ancestry. Now, is there research looking at where Crohn's happens around the planet? There is. I happen to work on tuberculosis primarily, and we benefit from a tremendous resource when we do epidemiologic tracking, which is tuberculosis is what's called a reportable disease. If you get tuberculosis, the government's going to have you in a register. And then we can look at your postal code, we'll know your age, sex, we're going to find out your contacts, we're going to know a lot more about you than you know, and almost as much as the NSA. But in the case of Crohn's disease, it's not a reportable disease. We don't actually know what is the incidence of Crohn's disease in St. Hyacinthe, what is the incidence of Crohn's disease in Saint Jérôme. That is done in more of an estimating way. So what do people do to estimate it? They say, well, infliximab is used for Crohn's disease, so we'll look at the doses of infliximab across the postal codes of Quebec. But we know it's also used in rheumatoid arthritis, and it's also used in some others. So there are some estimates of that, but it limits the ability to say what is the geographic dispersal of Crohn's disease if you don't have hard numbers of Crohn's cases. Another way to do it is to say, well, there are clinics that have Crohn's disease patients. So, Dr. Bitton and Dr. Cohen can say this is how many Crohn's patients they have, but they're magnets, right? And we know from the healthcare system, the people from Laval are being told not to go to the Jewish General Hospital. So are they a Laval postal code person or are they a JGH clinic person? So the denominators get a bit wobbly. But when you look at those clinics and you look at those reports, you do get a sense there's a sort of a global atlas of Crohn's disease, and Canada seems to be among the highest incidence, if not some of the highest incidences ever reported. Quebec has an incidence of about 20 per 100,000 and a prevalence of about 400 per 100,000, and that has only been equaled to my knowledge in New Zealand. Other countries have slightly lower incidence. So then you say, is it a north-south thing? Well, Canada is north and New Zealand is south, so I don't think you can then simply say, it's in the north. There is a map of Crohn's disease prevalence that was published a few years ago by a Greek author in the journal IBD. And I took the incidence from the 26 countries that he reported, and I put it in this extremely sophisticated software program called Excel. And then next to that, I went and got the GDP per capita for each of those countries, and the incidence of cardiovascular mortality for each of those countries, and the incidence of tuberculosis for each of those countries. And the highest correlation was inversely between Crohn's and tuberculosis. There was a very strong correlation coefficient in a negative sense. The high Crohn's were low TB, the high TB were low Crohn's. There was more of a scatter when you looked at things like income or cardiovascular disease. Does that partly answer the question? Yeah.
There was a question here, then let's take three more. 1, 2, 3, yes.
Yeah, and then we'll stay around for coffee after. So we'll take three and then, okay.
Yes, please.
Before you were saying that after the cow, once the cow is very sick and can't produce milk anymore, it's slaughtered, okay? What happens to the cow once it's slaughtered?
So the question was, I'm just going to repeat, if a cow is very sick and it is sent for slaughter, what happens to the cow?
Yeah, because the population at large is really eating sick meat.
That's right. So there's a MAP researcher in Wisconsin who says that the current farm practice is called test and cull. If you test positive, you kill the animal. That was used also for bovine tuberculosis before. That's how we got rid of bovine tuberculosis from the farms. And he says that people are unaware that what test and cull means, this is Mike Collins, really means test and feed it to the public. So it is possible that hamburger has MAP. And it is possible that some of the products in the grocery store have MAP, just like we know that some of the products in the grocery store have listeria and they have E. coli. What we do not know is when you go from the farm to the fork, we don't have a very good understanding of what happens next. MAP is a bacteria that only lives inside of a host. So if you just were to leave it on a shelf at a grocery store, the numbers will not proliferate like some of those other things that cause foodborne diseases. In fact, you'd expect it to gradually decline. We don't know what a naturally infective dose for humans would be. If I gave people in the room a liter of water here and I said there are 2 bacteria in here, would you be afraid? 10 to the 2, 10 to the 4. That has been done for Shigella. That has been done for E. coli. We've worked out the infective dose of typhoid for people using volunteers. We have no idea if 10 to the 2 or 10 to the 4 or 10 to the 6 MAP that might be in ground beef is or is not a risk. And that becomes very difficult because people want to say, well, if MAP is on the farm, meat is a problem, or if MAP is on the farm, milk is a problem, but we don't actually know. As some of you know, E. coli 0157 also comes from cattle. And people want to say, oh, wow, that's hamburger disease, but you probably also know E. coli 157 has contaminated vegetables and salads. And so therefore MAP could be in vegetables and salads, and I really don't know, and there's not a lot of research on it. And you know the reason there's not a lot of research on it, because the MAP driver of research on the farm is the economic problem on the farm, and whether or not it causes human disease is a critical link to ask those questions. But until we know if it's linked to human disease, nobody has the resources to formally identify answers to those questions. They're very tough questions.
There's been a lot of done.
There's a lot of research on whether I take 10 liters of milk, I can find MAP in it.
Right.
There's not much research telling me if 10 MAP or 100 MAP or 1,000 MAP is a problem
Yes, there is.
in a human.
Yes.
Okay, well, maybe over coffee you can show me the literature where somebody has challenged people with
Well, I
10,
can send it to you
100,
by
and
email.
1,000.
I
Certainly,
don't have it here.
because I am unaware of any human challenge where somebody's done a dose escalation of MAP and found out what is or is not an infectious dose
There
for
is
people.
that information.
Thank you. I look forward to that. Thank you.
There was a question here and then one back here. Yes.
Oh, yes.
Do you believe that there's a link or that Accutane could be a trigger to Crohn's disease?
It is not my field of research.
What's the question?
The question is there a link between Accutane and Crohn's disease? It is not my field of research. I do know a very well-respected gastroenterologist in Winnipeg named Charles Bernstein, who set up, partly in response to your question, sir, about what is the incidence of Crohn's disease, he set up one of the first places where he created a province-wide registry. So he knew everybody with Crohn's in Manitoba, and he could look by postal code and by county at all the incidents, and by having all that information, it enabled him to ask questions quite rigorously, like, is there an association between Accutane and Crohn's disease? And his literature, his paper said no. He could not find a signal. There was no association in his paper. I don't have the reference right now in my mind. If you send me an email, I could certainly send you the reference, but it's from Charles Bernstein, University of Manitoba, and he's a very respected Crohn's epidemiologist.
Final question? Yes.
Just to expand on the question that was asked geographically about Crohn's disease, do you have any information on why the incidence rates are sort of have been higher in more modern times, let's say like in the last 5-10 years versus 20 or 30 years ago? Or is it just we're collecting better data now?
Sure. So the question is, why is the incidence of Crohn's disease in many studies rising, and what could be the drivers? And you know, I'm almost speaking out of turn when I'm being asked, how do gastroenterologists diagnose Crohn's disease? How do they report Crohn's disease? How do they treat Crohn's disease? I've asked gastroenterologists this question, I've asked surgeons this question, so partly this is secondhand information. There's undoubtedly, when you recognize a disease, you diagnose it more, and when you diagnose it more, you recognize it more. And especially when you go from the 1930s when gastroscopy and colonoscopy were not available to the 70s and 80s, you're going to have some sort of a surveillance effect. So that if you are thinking of it more and you are better able to diagnose it, the numbers will go up. If you were to go and take healthy, otherwise well people and put capsule endoscopy in all of us, you might find little lesions in a few people. And you would then say, does this person have Crohn's disease, or does this person have a self-limited little thing that would have gone away otherwise? So I think that there's some sort of a surveillance issue that has to do with recognition and capacity. But I think there's also something real. And most people are saying that beyond that, there is a biological rise in Crohn's disease, and that the biologic rise in Crohn's disease is too fast to assign Crohn's to being only a genetic disease, because our gene pool is not replacing and changing fast enough to say that we have twice as much Crohn's in 2010 as we did in 1990. So that would suggest that there are environmental factors that are also predisposing to Crohn's disease. In the model that I present here, you would call that sort of a two-hit model, where you'd say, well, if you are genetically susceptible and you are infected, together they could conspire to be disease. But there's also going to be modifiers. We've shown in a paper with John White's group, and Ernie Seidman was an author of that as well, that vitamin D is a modifier of the NOD2 pathway. So that when NOD2 is broken, vitamin D doesn't work as well, and therefore you can't get the benefit of the immune protection of vitamin D. We know that the birth control pill and cigarette smoking are also risk factors that modulate the risk of Crohn's disease. So just because something's going up doesn't mean forcibly that the bacterial trigger is going up. It could be the bacterial trigger is more or less the same, the genetic risk is the same, but the modifiers could be changing, and then you get into those situations that some of you might remember from high school math when you're trying to solve the unknown, and you realize that if there were more unknowns than there were equations, you were going to kind of get stuck, and you either had to find more equations or you had to solve some unknowns. And I think we need much more information before we can actually solve that. But I believe that there is a real rise as well as also a surveillance rise, and together that makes it even trickier to dissect out.
We're done. Thank you, Marcel. You've all heard, I think, a remarkable lecture tonight. If you hear from medical students about autoimmunity, and I see many of them over time, they will tell me that Crohn's and ulcerative colitis are autoimmune. That lupus and rheumatoid arthritis is an autoimmune. They're not. There are only 3 autoimmune diseases that we can actually count on. Type 1 diabetes, thyroiditis, and something called myasthenia gravis, where we know that the cause of the disease is autoimmune. Lots of people have antibodies that do nothing. But that's either here nor there. So you've learned a great deal tonight. And every one of the things that Marcel has told you can be expanded into a full lecture series, I promise you. But I think you have the knowledge. I think you've probably heard about a remarkable model, which I think is going to give us enormous information in the future. But obviously you go from cause to effect to cure, and that takes time. Thank you.
Thank you very much.